Our topic today is one of the topics of lipid metabolism. We're going to talk about ketone body metabolism. This topic includes the sources and phase of acetyl-coenzyme A, which is a precursor substrate for ketone body synthesis. There is also explanation for the sense and degradation of ketone bodies, as well as regulation of ketogenesis and ketolysis. The ketone bodies are compounds or acidic compounds produced during the metabolic conversion of acetyl-coenzyme A, which is derived from the beta oxidation of fatty acids, the pyruvate dehydrogenase reaction after finishing glucose oxidation that ends in pyruvate, which is converted to acetyl-coenzyme A by the pyruvate dehydrogenase reaction. Also from the carbon skeletons of amino acids during their catabolism. This precursor substance, when increased, it is channeled to the liver cells, and the ketone bodies are synthesized in liver cells. Ketone bodies are synthesized from a substrate which is acetyl-coenzyme A, which is produced from different sources. One of these sources is the beta oxidation of fatty acids, or the carbon skeletons of ketogenic amino acids, and also from carbohydrate metabolism, or precisely from glucose oxidation, which ends in pyruvate that is converted to acetyl-coenzyme A by the pyruvate dehydrogenase enzyme. Ketone bodies are acidic products, and they are normally produced in the body, but their synthesis increases when there is increased presence of their substrate, which is acetyl-coenzyme A, as we mentioned, that it is a product of fatty acid oxidation during periods of starvation and prolonged fasting. The acetyl-coenzyme A produced normally is channeled to the citric acid cycle, where it combines or condenses with oxaloacetate to form citric acid or the tricarboxylic acid that fuels the citric acid cycle, ending in production of energy, carbon dioxide, and water. We have three compounds, namely acetoacetate, β-hydroxybutyrate, and acetone. These are the three ketone bodies produced in the body. The synthesis of ketone bodies or ketogenesis occurs in the liver mitochondria, and as we mentioned, this occurs normally from acetyl-coenzyme A, which is a precursor substrate that is derived from beta oxidation of fatty acids. And during periods of prolonged starvation, there is excess mobilization of tissue lipids and lipids in the liver, with the resultant elevation of free fatty acid production and beta oxidation of fatty acids. The result of beta oxidation of fatty acid is the production of acetyl-coenzyme A, which is a substrate for ketone body synthesis. The overflooding of the liver with free fatty acids and acetyl-coenzyme A resulting from their oxidation exceeds the liver capacity to channel these substrates to the citric acid cycle to be utilized. And there is also depletion of the oxaloacetate, which is used for gluconeogenesis during periods of starvation. So the Krebs cycle will be slow due to the deficiency of the substrate, which is oxaloacetate. The steps of ketone bodies synthesis starts with a condensation of two acetyl coenzyme A molecules by the enzyme thiolase with release of coenzyme ASH, and the produced intermediate is acetyl coenzyme A. Condensation of a third molecule of acetyl coenzyme A by the help of hydroxymethylglutarate coenzyme A synthase results in the formation of a major product, which is a beta-hydroxy beta-methylglutarate coenzyme A or hydroxymethylglutarate coenzyme A. This compound undergoes a lyase reaction by hydroxymethylglutarate coenzyme A lyase with release of acetyl coenzyme A and the production of the first ketone body, which is acetoacetate. Acetoacetate undergoes a non-enzymatic conversion to acetone and may undergo reduction by beta-hydroxybutyrate dehydrogenase to form beta-hydroxybutyrate. So by the end of this synthetic process, we have the acetoacetate, beta-hydroxybutyrate, and acetone, which are the three ketone bodies formed in the liver. It's important to remember that during periods of starvation, the liver starts the process of gluconeogenesis, and the gluconeogenesis utilizes the oxaloacetate, which is a substrate or one of the substrates of the Krebs cycle. So when gluconeogenesis starts, there is depletion, or there would be depletion of the oxaloacetate and slowing of the Krebs cycle, and there will be reduced consumption of acetyl coenzyme A and the citric acid cycle, and this is going to lead to overflooding of the liver with acetyl coenzyme A, which is also increased by increased fatty acid oxidation as a result of the prolonged starvation. So the question here is, why do we synthesize, or the liver synthesize, ketone bodies? During periods of starvation and or prolonged fasting, there is great need for energy, especially by the brain tissue. The liver then starts to perform the process of gluconeogenesis, but when the substrates for gluconeogenesis are depleted or reduced, the liver starts to catabolize fatty acids, producing acetyl coenzyme A. Excess acetyl coenzyme A is incorporated in the process of ketone bodies synthesis, which are transported after synthesis, and the liver mitochondria are transported to the blood, then to the tissues that need energy, which are mainly the brain and skeletal muscles. Also, the process of ketone body synthesis is responsible for generation of the free coenzyme A, as we mentioned, that is required for the process of beta oxidation. So we have a question here, is why it is obligatory to synthesize ketone bodies. First, there is energy deficiency, especially for vital organs like the brain. Second, there may be conditions of reduced insulin production with consequent defect in carbohydrate metabolism, and hence the body is forced to utilize fatty acids after activation of the hormone-sensitive lipase in adipose tissue, with release of free fatty acid in the blood prior to their oxidation. Then the liver starts to oxidize these free fatty acids, producing the substrate for ketone body synthesis, which is acetyl coenzyme A. So the sequence of events will be, when starvation is prolonged or fasting is prolonged, there is relative or absolute insulin deficiency, then there is mobilization of free fatty acid from adipose tissue by lipolysis, followed by increased delivery of free fatty acids to the liver, where they are oxidized after they are uptaken. Their oxidation yields acetyl-coenzyme A, which exceeds the liver capacity to channel these acetyl-coenzyme A molecules to Krebs cycle. So there is acceleration of ketone body synthesis to get rid of the increased amounts of acetyl-coenzyme A. What is the fate of these ketone bodies after synthesis? As we mentioned earlier, when there is high rates of mobilization of tissue lipid, free fatty acids pass to the liver, where they are oxidized to produce the active acetate or acetyl-coenzyme A, which acts as a substrate for ketone body synthesis. And this occurs during long-term fasting or starvation, or in case of uncontrolled diabetes, where there is relative or absolute deficiency of insulin. These processes, especially the process of increased fatty acid oxidation, produce sufficient or excess amounts of acetyl-coenzyme A that undergoes ketone bodies formation in the liver mitochondria. Ketone bodies are acidic compounds, but they are preferred fuel for some cells, especially the brain cells, skeletal muscle cells, kidney, and intestine. These ketone bodies are preferred even if glucose is present. So they are used by the brain cells, but they are not used by liver cells. But how ketolysis occurs? As we know that β-hydroxybutyrate is one of the ketone bodies utilized by the brain cells or skeletal muscle cells. Ketolysis occurs first with oxidation of β-hydroxybutyrate by the β-hydroxybutyrate dehydrogenase enzyme, with release of reduced NAD. This process of oxidation yields acetoacetate that undergoes a thiokinase reaction with production of acetoacetyl-coenzyme A. Acetoacetyl-coenzyme A undergoes a thiolase reaction, cleavage, with release of 2 units of acetyl-coenzyme A that are channeled to Krebs cycle to be utilized for energy production, especially in brain and skeletal muscle cells. So this process is a reversal of ketogenesis that occurs in the mitochondria of these cells. The utilization of ketone bodies by extrahepatic tissues occurs when the levels of ketone bodies in the blood exceeds 3 milligrams %, and this occurs with prolonged starvation after about 3 days. The levels of ketone bodies start to increase, and this process is in favor for the tissues that have the enzymes that can oxidize ketone bodies, like the brain cells or skeletal muscle cells, which have the enzymes that can oxidize the ketone bodies and utilize them. And these enzymes are induced, especially succinyl-coenzyme A and acetoacetyl-coenzyme A transferases, which are induced with increasing levels of ketone bodies. And this allows the brain tissue to utilize ketone bodies as a source of energy. The liver does not have these enzymes, so it cannot utilize ketone bodies. So the liver can synthesize but cannot utilize ketone bodies. And this will end the process of ketone body synthesis and ketone bodies oxidation. If you like this lecture, please hit the subscribe button and give your feedback in comments. And you can find other details for this lecture in the website provided.